Today I'm going to be talking about the identification of the transcription factor TBET in channel catfish. Th1 cells, or T helper cells, are lymphocytes that participate in adaptive immunity. These cells are a specific subset of CD4 positive T cells, and they are important in the protection against intracellular pathogens and viruses. These cells produce interferon gamma, interleukin 2, and TNF beta, which induce cell-mediated immunity by the activation of cytotoxic T cells, macrophages, and NK cells, or natural killer cells. These cells are activated to kill tumor cells, virus cells, and cells infected with intracellular bacteria. TBET is a transcription factor found to be associated with the T helper 1 cells in mammals. And TBET has been shown to control the expression of the Th1 cytokine, interferon gamma, which activates cytotoxic T cells and macrophages. Previous studies in channel catfish have shown that TBET cells express messenger RNA for TCR beta and alpha, the T cell co-receptors CD4 and CD8, and the T cell growth factor interleukin 2 and INF gamma. Based on the information that we know so far about catfish T cells, we hypothesize that catfish have distinct T cell subsets, which are controlled by the expression of signature transcription factors, which is TBET in the case of Th1 cells. So first, in this project, the first thing we did was perform reverse transcriptase polymerase chain reaction, or RT-PCR. And to do that, we first extracted the total RNA from catfish leukoid tissues. Next, we treated it with DNase and made cDNA from 2 micrograms total RNA using superscript 3 reverse transcriptase. Then we amplified a partial TBET sequence using primers complementary to catfish VSTs with homology to zebrafish TBET. I ran the PCR products on an agarose gel, and as you can see, TBET is expressed in all the tissues that we analyzed, including gill, intestines, spleen, head, kidneys, and trunk kidney. The spleen PCR product was cut out of the gel and purified. I then cloned the PCR product into PCR4 topo and sent it off for sequencing. In this figure, we have an alignment of the deduced amino acid sequence of catfish TBET with zebrafish, salmon, and carp TBET sequences. The identical amino acids are shaded in red, and as you can see, the catfish sequence is very similar to the TBET sequence in other fish. Not only do we look for the presence of TBET in catfish tissues, but we also look for it in peripheral blood lymphocytes and clonal catfish T cell lines. To do this, RNA was isolated from the PBL, or peripheral blood lymphocytes, and from clonal catfish cell lines. RT-PCR was again performed using primers specific for TBET, TCR beta, and MIGM. TCR beta and MIGM were the positive control groups in this study. And as you can see, PBL and 1 clonal T cell line were found to express TBET. Additional experiments that could be performed in this area would be to isolate the full-length TBET cDNA using 5' and 3' race. And at this point, we believe we have isolated the 5' and 3' race products, and we've sent them off for sequencing. Another thing we could do is to determine if TBET controls the expression of interferon gamma in catfish. I would like to thank the Catfish Immunology Lab for their help this summer. Without whose help, I would not be able to do this. Those people are Dr. Binkton, Dr. Wilson, Aaron Taylor, Cecile Snell, and Thomas Rousseau. Thank you.
Change the program. Mohammed Al-Daklala